We're very honored to participate in the Galien Forum. I want to just apologize. Joel Marcus is not with us today. He's sick. So my name is Lynne Zydowsky, and I will moderate the panel. And the title of the panel is Autism, the Next Decade. So just to start with a few words about autism spectrum disorder. Autism affects 1 in 68 children, approximately 3 million people in the U.S., with a cost of $38 billion. Those costs include healthcare, education, ASD-related behavior, family services, and caregiver time. Autism now encompasses a single diagnosis of autism spectrum disorder and is characterized by impairment in language, social interaction, and restrictive and repetitive behaviors. The average age of diagnosis is 4.5 years, yet the average age of first concern is 19 months. Less than half of all children are diagnosed by the time they are 3 years old. I'd like to read a quote from Geraldine Dawson, one of our panelists. "We do not have to wait until we make a reliable diagnosis to begin intervention. If we can help a baby become more engaged and interactive and develop gestures and language, we can possibly prevent autism. As we know today, being able to diagnose in that first year is imperative to starting early behavioral treatment." ASD is a very heterogeneous disease, and to date over 200 genes have been identified and implicated, but basic research is still needed to understand the molecular basis of the disease and to identify targets for intervention. We've seen in the last few years several attempts to develop a treatment for Fragile X syndrome, which is a single gene disorder which accounts for 5% of all autism cases, using this as a strategy to develop treatments for disease. However, while drugs may correct the synaptic abnormality and even enhance learning, it may not address behavior, and it will most likely take combinations of pharmacologic treatments with behavioral intervention and multimodality approaches to effectively treat this disease. With our esteemed panel today representing this diverse set of perspectives from the community of people working on ASD in research, clinical models, drug discovery and development, patient care, and advocacy, we will discuss how one can translate breakthroughs in medicines, genetics, and clinical trials, as well as the increasingly critical intersection of science and technology, into advances that will improve the quality of life and hopefully one day treat ASD. Today we'll discuss the most recent progress in treating autism spectrum disorder. So I'll just start by introducing the panel. To my left, I have Geraldine Dawson, who's the professor in the Department of Psychiatry and Behavioral Sciences and director of the Duke Center for Autism at the Brain Development at the Duke University School of Medicine. Dr. Dawson is president of the International Society for Autistic Research. She is a licensed practicing clinical psychologist and scientist with more than 225 published papers and 10 books on early detection and the treatment of autism and brain development. Her scientific research has been recognized by the NIH as one of the top 10 advances in autism research from the years of 2007 through 2013. To the left of Jerry, I have Dr. Rob Ring, who serves as the Chief Scientific Officer of Autism Speaks. It's the world's largest autism science and advocacy foundation, and over the past decade, they have invested over $280 million into basic and applied research globally. He's a neuroscientist by training. He's responsible for shepherding the science mission of the foundation and overseeing a diverse portfolio of research investments aimed at improving diagnosis, treatment, and prevention of autism spectrum disorder. To the left of Dr. Ring, I have Dr. Santarelli, who joined Roche in 2005 as the CNS Discovery Group Leader. Luca established the Neuroscience Clinical Regulatory and Exploratory Development Department, focusing on translational neuroscience, clinical proof of concept, and novel therapeutics for psychiatric and neurological disorders. And in 2010, he became the global head of Neuroscience Disease Translation Area. One of his key achievements in this position at Roche was the creation of the industry-leading R&D department to discover and develop drugs for neurodevelopment disorders such as autism and Down syndrome. And to the left of Luca, I have Laura Slatkin, who began her career on Wall Street at Lehman Brothers. And following her successful career there, she joined forces with her husband, Harry Slatkin, to found Slatkin & Company, a luxury home fragrance company. In 1999, shortly after giving birth to twins, the Slatkin son was diagnosed with severe autism. In response to this life-altering disease issue, Laura and Harry, along with a group of colleagues, founded New York Collaborates for Autism, an organization dedicated to autism education, community outreach, and biomedical research. They founded the first public charter school dedicated to serving children with autism, called NYC Autism Charter School, which opened in 2005, serving as a model for the New York City public schools. In the fall of 2008, they opened the doors of the Hunter College Autism Center to train at a graduate level professionals to serve people with ASD. And their largest initiative to date has been a partnership with New York Presbyterian Hospital and its affiliated med schools at Columbia and Weill Cornell to form the Center for Autism and the Developing Brain. And with that, I'd like to ask the panelists each to give a few minutes of discussion about their current focus in the area. And once we complete that, we'll open up to some questions with the panel and to the audience. Thank you.
Good morning. Make sure this is. Well, thank you so much for having me. This is an amazing forum. I had to prepare for the panel, and I had to pull myself away from the first set of presentations, which were so exciting, and I keep thinking about how those will apply to autism someday, I am sure. So it's terrific to be here. As Lynne was saying, autism spectrum disorder is a heterogeneous condition. In fact, it's not one condition, it's many conditions with many different causes. When you think about a typical baby and the early development of that baby's brain, babies come into the world wired to be interested in people. You don't teach a baby to make eye contact or to vocalize. Our brains are really wired so that we notice that people are very special and we pay attention to them. The baby with autism, somewhere between 6 and 12 months of age, changes course in terms of that tendency to engage with the social world. And that baby becomes much more interested in objects, doesn't pay attention to people. What this does is it reduces the opportunities for that baby to engage in interactions that are really essential for learning about faces and expressions and language and social interaction. And this further impacts brain development. In fact, what we may see in terms of autism as it's diagnosed at age, say, 2 and 3, is really the secondary impact of having spent a very important period of early development failing to engage in the social world and not getting adequate stimulation to those neurocircuits that underlie social and language development. Now, we're at a very exciting point in terms of the history of treatment development for autism spectrum disorders. Through very sensitive behavioral interventions, it is possible to draw the baby's attention back to the social world and to stimulate those neural connections that otherwise would not have formed. Research has shown that early behavioral intervention can alter the course of not only behavioral development, but also brain development in children with autism. So children who receive early intensive behavioral intervention have, on average, a 17-point increase in IQ. About 50% of children will show at least a 1 standard deviation increase in their IQ, which is quite dramatic. Moreover, we've now shown that this alters brain trajectory development and that the brain responses to social information is now much more like that of a typical child. In fact, in some cases, indistinguishable. So a major challenge now is getting access to these interventions. As you heard from Lynne, the average age of diagnosis is 4 to 5. And so this is really the gap between the science and what we can implement in the community is huge. And that is a major challenge that will be addressed to a large extent with things like insurance reform, but also through technology. Now, the beginning signs of autism can be recognized very early in life, even before the onset of the behavioral symptoms. Now, this is really important. If you think about areas like Alzheimer's or Parkinson's, you know how important it's been for those fields to be able to think about treatment before we have full Alzheimer's disease manifest. And similarly, this is the way that we're thinking about treatment in autism. So what we've learned so far is that in the second half of the first year of life, there are differences that can be detected in white matter development using MRI and diffusion tensor imaging. And we can see that the patterns and ways that the brain is being connected, particularly long-range connections with the frontal lobe, are not developing normally. We also can detect early signs of autism using EEG and measures of electrical activity, and see differences there that then precede the onset of the syndrome. We also see very early changes in patterns of attention, as I mentioned, where the baby is no longer paying the normal kinds of attention to social information. So current trials are actually beginning the behavioral intervention when infants are at risk for autism between 6 and 12 months of age. We're just reading out a clinical trial from the University of Washington where babies were randomized into treatment versus non-treatment at 6 months of age by virtue of the fact that they were at risk for autism. And we're able to show that we can change the course of development, both in terms of behavior and brain development early on. Now, although great strides have been made in early behavioral intervention, we also know that many individuals with autism remain very impaired despite having received high-quality behavioral intervention. To give you a sense of this, about 30% of people with autism never speak. They're minimally verbal. And many of those individuals did receive very good intervention. So we know that medicines are needed that would enhance neuroplasticity and improve outcomes for all individuals. But yet, currently, we have no FDA-approved drugs that actually address the core symptoms of autism, which are social communication impairments and repetitive behaviors. Because of investments over the last couple of decades in genetics and neuroscience, though, we know much more about the molecular and cellular basis of autism. What we've discovered is that many of the autism genes converge on a few major signaling pathways, and we'll hear more about this from the other panelists. And that it appears that a pattern of dysregulation between inhibitory and excitatory neurotransmitters at the synapse are consistent across several animal models of autism. And this has inspired pharmaceutical companies such as Janssen and Roche, which you'll hear about, to develop and test medicines with the goal of increasing neuroplasticity and reducing the core symptoms of autism. A major challenge, however, has been how to optimize success in clinical trials. We absolutely need validated biomarkers that can be used to stratify samples because we know that autism is a biologically heterogeneous disorder. We need early efficacy biomarkers that can quickly determine whether a drug has reached its intended target, and we need much more sensitive outcome measures that will allow us to determine whether a treatment has actually worked. These are challenges that are being currently addressed in two major efforts. First, in Europe, and you'll hear more about this from Luca Santarelli, is as part of the Innovative Medicines Initiative, there's an international consortium of scientists led by Roche and King's College London that has launched the largest ever research academic industry collaboration to find new methods of development of drugs for autism spectrum disorder. And a large focus of this is on discovering biomarkers that could be used for stratification. In the U.S. this year, the Autism Biomarkers Consortium for Clinical Trials was launched. This is a $34 million project that's funded by the NIH in collaboration with the Foundation for NIH, the Simons Foundation, Janssen Research and Development. The project involves 5 universities, so Yale, Duke, Harvard, University of Washington, and UCLA. And the goal is to validate 3 biomarkers. One is DNA, the second is eye tracking, and the third is EEG, and with a particular focus on stratification for clinical trials. Some of the methods that are being used are shared with the project that is being conducted in Europe, so that we'll be sharing data across the US and Europe. And then finally, Janssen Research and Development has invested in a very novel platform for measuring outcomes in autism pharmacological trials. We're involved in validating this platform at Duke, and several other universities across the United States are also involved in this project. And the platform uses biosensors and very innovative tools for assessing autism symptoms in the home environment. So over the next few years, we hope to have a panel of relevant biomarkers that will provide a unique profile for each person with autism and allow more precise clinical trial designs. These clinical trials are very likely to combine both medical and behavioral treatments. So I hope this gives you a sense of both the hope and the progress that we're making in developing more effective treatments for autism.
So Rob.
Good morning, everyone. Thanks, Lynn, and thank you to the Galien Foundation for the opportunity to participate this morning. I think you guys are in for a real treat once we get into the discussion, and so I don't want to really delay that too much. And what I'd like to do is just offer a few comments on things that we're doing at the foundation at Autism Speaks that are aimed, strategically to try to move and address bottlenecks in this value chain of turning the many breakthroughs in science that you're going to hear about today into actual things, applications, diagnostics, medicines, guidelines, you name it. And I'd like to touch on a couple of what I see as sort of fundamental challenges facing the entire research ecosystem in autism as we tee up the rest of the conversation. We've already, Lynn did a nice job in introducing what I do at Autism Speaks. I do benefit in some ways, having led the autism unit at Pfizer before joining Autism Speaks, to see this space from a number of different angles, and it's been a real pleasure to be working in the nonprofit world on this really incredible, important problem. At Autism Speaks, we have, beyond our traditional areas of research funding, started to work beyond the science. We've been traditionally an advocate in Washington and have had our fingerprints over major legislation like Autism Cares, which was passed just last year, appropriating over $1.3 billion of new funding to HHS for autism research and services across HHS, and that's a significant chunk of support for the field. We've been looking for ways to leverage the momentum we have in Washington, not just through the initiatives such as 21st Century Cures and Precision Medicine Initiative, et cetera, that are ongoing, but looking for ways to help move this story forward in the regulatory area, and that's, I think, a really key part of this story that doesn't get talked about enough. Building off of everything that Jerry just said, which I think was a wonderful summary of what the state of autism really is right now. As a foundation, one of our key investments, or key plays, if you will, has been a project called Missing, which is spelled MSSNG. I encourage you to take a look at the website. This is a collaborative partnership we initiated with Google over 2 years ago, and the goal of this project is to build an open access database of whole genomic sequence data on individuals with autism and their families, on multiplex families. By the end of first quarter next year, we will have completed the sequencing, the whole genome sequencing, not exome, the whole genome sequencing of 10,000 individuals and their families. We've already crossed 5,000 mark, and our portal is up and running. And really our central thesis, if you will, around this is builds off years of research that have indicated that when it comes to understanding the risk for autism, although we can't deny the role of environment and other factors, many of the signs point to the role of the genome. And the genome may be an incredibly important mechanistic explanation for how the environment is working in autism as well. And we think in the genomes of these 10,000 individuals and what gets built beyond that lie the keys to understanding diagnosis, reducing the complexity of that heterogeneity that we all recognize in autism, and really will be that handle for target identification and rationalization for the future. And it's really an exciting time for open science, period. I think as we look at the opportunities in the autism space, there are a number of different efforts currently getting underway. The Simons Foundation has a project called Spark that is attempting to enroll 50,000 individuals with autism and build a database around that. I think this convergence of technology and biological sciences, life sciences, is going to be will play a significant story in the autism space. As I'll wrap up here and just make way for discussion, a couple of points I think are really key and will be a theme that will dominate a lot of research moving forward. One in particular is recognizing that autism, although diagnosed in the first years of life, is a lifetime diagnosis. And there's a lifetime of challenges and opportunities for individuals on the spectrum that we don't fully understand. And we have to expect that the addressable, targetable biology for medicine's development will alter throughout the lifespan as well. So even in the context of medicine's development, we're not just developing product concepts for adolescents and youth, but there's an entire lifetime of challenges that have to be considered. I'd also just like to echo something that's already been said multiple times. I can't emphasize it enough, but the average age of diagnosis hovering over 4 years is unacceptable. It's especially when we know we have the means to diagnose autism much earlier than that, and really the opportunity cost there for individuals and families are losing out on the access to early interventions that we know have profound effects on outcome. I think this needs to be a key focus of research, and I'm sure we're going to be talking about a number of things related to that. So look forward to the discussion, and I'll make way for Luca.
So first off, I'd like to thank Lynne and the Galien Forum for putting autism on the agenda. This is always a topic that excites me, as having been the person that has championed this at Roche as an effort in R&D in our organization. So I'd like to spend the next 5 minutes telling you about why we started working on autism in a big pharma company like Roche, and how we went about setting it up and how far we got this far. It all dates back about 5 years, where we reasoned that the advances in genetics had provided enough evidence about what targets to work on, so that autism had become tractable from a drug discovery, drug development standpoint. That we could conceptualize a rational R&D effort at Roche. And again, the key aspects of that effort were how do we choose targets, how do we create adequate translational models, in vitro, in vivo translational models, and how do we find the right patients? How do we sort of resolve the complexity and heterogeneity of the clinical presentation of this condition and find those that are more likely to benefit from our treatments? And I'll start from the target. So as I said, genetics had shed a lot of light on some of the key pathophysiological aspects that underlie autism. We Roche initially focused on those types of autism, syndromic autism, that are found in single gene Mendelian forms of neurodevelopmental disorders, such as Fragile X mental retardation, tuberous sclerosis, and in some cases in other types of genetic conditions that are not single gene, but like Down syndrome, for example. So these are the space where we first look for targets. In addition, science had evolved to demonstrate that there were some discrete circuits in the brain that played a role in the modulation of some of those behavioral dysfunctions that are found in autism. For example, social behaviors. And there we particularly focus on the vasopressin oxytocin system for all the work that had been done in the past 20 years, demonstrating that social behaviors can be modulated by those peptides. At the circuit level, we also focused on another system which came out of research in the area of Down syndrome. And that was the balance between excitation and inhibition in the brain. It became apparent about 5-6 years ago from studies in a mouse model that reproduced the trisomy of chromosome 21 in a mouse, which is a trisomy of chromosome 16, that an excessive GABAergic tone was likely to cause some of the cognitive dysfunctions that are found in this condition. So we went both a molecular target approach but also a circuit target approach for the choice of our early targets. In terms of translational tool, we really focus on two levels, cellular and in vivo model, animal model level. And again, what we did, we engineered into other cell systems or animal model system, primarily mice, those mutations that were known to cause either syndromic autism or very rare types of autism, such as the mutation in neuroligin gene, orexin gene, UB3A gene, and created a whole set of a toolkit with both cellular systems, so in some cases derived by patient using the iPSC technology, in some cases engineered by ourselves, that enable us to do both a cellular and an in vivo all animal analysis. So we had to recreate a lot of the animal models that, or create from scratch a lot of animal models that didn't exist at that point. But perhaps the hardest thing was the third challenge, which was identifying the right endpoints in order to measure the activity of our drugs in patients and identify what subset of patients we wanted to study first, depending on the agent that we were examining. So in that case, we really resorted to the whole scientific community in terms of expanding observational studies or leading the charge in the creation of large observational studies, multicentric observational studies. And it was mentioned the EU aims is one of these. One of the objectives of the EU aims was to create a large study that enable us to study longitudinally patients with autism, profile them both genetically, molecularly, but also deep phenotyping using certain circuit approaches such as functional MRI or EEG to look at the brain in a more functional way. And we did that on a large scale in the case of the EU aims, but we also did it on a smaller scale in some internally designed observational, non-interventional studies in Down syndrome, for example. So I can give you an update on the EU aims, but we're starting to see the first results. We're starting to have first evidence that we could actually use, for example, EEG in clinical trials to determine whether a patient has the right level of or alteration in his pattern or electrical activities. We can start using functional MRI in response to look how the brain activates in response to socially relevant stimuli, and these things can become more quantitative means to assess drug effects. And lastly, we really focused on adapting our trials to those patients we were trying to treat depending on the mechanism. So far, we've experienced with 3 programs which are in the public domain. One is the Fragile X program, which we approached with an agent which is called is an mGlu5 antagonist. And here, what we try to do to create a more homogeneous patient population. This is a genetic single gene condition, but still very heterogeneous because the level of changes, and even at the single gene level, it varies depending on the methylation of the gene. So we really try to create more homogeneity by selecting a particular number of variations of triple nucleotide repeats in that patient population based on level of functioning, so deep phenotyping, based on level of severity, age, gender, all of these factors were utilized to reduce heterogeneity. In the case of, we had a second program with a GABA alpha 5, a very selective GABA inverse agonist in Down syndrome, and in that case, really we focused on those patients where we thought there was evidence of excessive inhibitory tone, and then again age stratification, severity stratification. And lastly, the more recent program has been one in the vasopressin oxytocin system with a vasopressin antagonist, where we really focused on high-functioning autistic patients. This is the very first one in sporadic autism that we have embarked on, so again, we went into high-functioning, mostly high-functioning patients, where we could measure a social and functional impairment, again, age stratified, because you cannot take all comers in these trials. So, so far, the only study we have reported, which was negative, was the Angle 5 study in Fragile X syndrome. It's not only us, other companies have tried that. The question is, still, when you fail in these things, is it the target you picked, or is the design of the trial? So a lot of these questions still have to be answered. I want to mention another experience in this area done by Seaside Therapeutic with arbapelfen that has left some sort of a bittersweet taste in the mouth of all those that worked on it. Roche had an option to acquire the molecule, where there was evidence on some activity, but not sufficient to bring the molecule forward, at least in our eyes. But now the molecule has been licensed by the Simons Foundation, so there is a possibility to continue there. So this has been our experience so far, and it's only been 5 years, but I want to say we have really, I'm very hopeful that we will see great progress in the next years to come, and if we continue to perseverate in this area. Thank you.
Laura?
Good morning. Is this on? Yeah. Lynne kindly touched upon some of the programs that New York Collaborates has formed since its inception. She talked about our charter school that we started, and she also talked about Hunter College, our training institute that we built, and then our major brain institute that we formed with New York Presbyterian Hospital, Cornell, and Columbia Universities. And all of this work that we've done at New York Collaborates for Autism, and I also sit on the board of Autism Speaks, was done because of our son David, who was diagnosed with autism at 17 months of age. And David is part of the spectrum that Jerry had mentioned earlier, that 30% that doesn't talk. But of that 30%, then there's a part of that 30% that is very severely impaired by autism, which David is in. He not only does he not have language, but he's cognitively very impaired, and he also has aggressive behaviors. So even though he's received early intervention at age 17 months, he had 40 hours a week of early intervention executed by some of the finest educational experts on the Eastern Seaboard. Even though he received all that early intervention and great educational supports, he's now 16 years old, we made no progress. So even today, he still can't tell us he has to go to the bathroom, he's hungry, without getting very, very aggressive. So, in fact, David no longer lives at home. Now he lives outside of Boston in an adult program, and he comes home once a month, and just so happens I was sitting here thinking he just came home this weekend. And it was terrific to see David. We're always excited when he comes home. We had a terrific weekend. He came home Friday night. By the time we got to Sunday morning, he had bit his twin sister's arm, and he bit my leg really, really hard. It was time for David to go back to the New England Center for Children where he lives. So that leads me to the discussion of, if there's 1 out of 68 children that are affected by autism, what happens when they become adults? Where are they going to live? And that's the pressing issue that we're facing right now. So while we've opened the charter school and we've built these medical centers and, training institutes, now we're really talking about adults with autism and what are the proper supports and what are the proper programs that we need for this population. But it's not only autism. About a few weeks ago, I had the pleasure of listening to Tom Insel talk about mental illness. And he reminded us all that back in the 60s, the Kennedy administration passed a bill that closed asylums. They felt that that was not an appropriate placement for individuals with mental illness. And the idea was that they would close these asylums and they would build beds in the community with the proper support. Well, the terrific thing is that they did away with those asylums because they were bad places. People weren't getting better there, but they never built the beds in the community. So there's been a 90% reduction in beds for individuals with mental illness since the 60s. So the question is, where are those people today? And interestingly enough, if they don't have a strong family network to take care of them, they're either homeless or they're in our prison system. So that's kind of interesting when we stop to think about that. So when we look at today, the government is really clear. When we started thinking about, and New York Collaborates for Autism, when we started to think about building adult programs, housing in the community, the government is really clear that they don't want to fund institutional settings, going back to the decision made in the 60s. They want to support adults with autism living in the community with the proper supports. So we embarked on this major initiative to really think about adults with autism and what are the best supports and what are the best living programs that we could build. So when we think about supports for individuals with autism, the work that we have to do right now is not very dissimilar to what we did 25 years ago when we passed the American with Disabilities Act in 1990. And that was for individuals with wheelchairs. And what we did as a society is we changed the infrastructure of our communities, our buildings. We built on-ramps. We built widened entryways to buildings so people with wheelchairs could have easy access. We changed our bathrooms so that we could accommodate individuals with wheelchairs. We changed our infrastructure to accommodate these individuals so they could live in our and could work in our community. So that was really expensive. So when we think about individuals with autism, we think, what are the on-ramps for individuals with autism that we need to build in our community to support this population? So again, it's not as expensive as what we did for individuals with wheelchairs. It's really more about changing people's perception. So if 1 out of 68 individuals are affected by autism, we should see 1 out of 68 individuals with autism in our workplace, in our communities. We should be encountering them and accepting them and supporting them, whether we're at work or whether we're at leisure, recreational programs. So what New York Collaborates for Autism has done is we've built this new initiative called Neighborhood Networks of New York. And what we intend to do is build state-of-the-art residential homes, apartments scattered throughout our community with that are wired technologically smart homes. So they're state-of-the-art smart homes that are wired with a two-way communication system. That the homes, the apartments and the homes are wired with motion detectors, pressure pads, alarms, a central monitoring system, and we have trained staff that support these individuals with autism and provide that support when it's needed, indicated by this two-way central monitoring system, not just in case. So it's much less expensive to build these homes, these smart homes. And it's funny because it was at the Alexandria Institute when I was sitting next to Andy Conrad, and I talked about our program that we were building. He raised his hand as president of Life Sciences at Google Life Sciences, said, I want to build your smart home. So we've been chatting ever since and working together to build this new program. So our goal is to build these beds in our community, the same beds that were shut down in the late 60s. But the bigger issue really is mental illness. We've done a terrific job at raising awareness for autism. Pediatricians are now diagnosing it early. Teachers are raising their hand and flagging kids that are at risk for autism. But we also have anxiety, depression, schizophrenia, bipolar disorder. And people are really not talking about these illnesses. And doctors aren't catching it, teachers aren't catching it, and who's going to build the beds for those populations as well? So, we were talking earlier about how, the new brain initiative really is going back and saying, it doesn't really matter whether you have autism, bipolar, depression, schizophrenia. Rob Ring and I have discussed the fact that it's something that's gone wrong with the brain. And so it's interesting to me how, when I look at all the different mental illnesses and the disorders, we're all facing the same issues. How are we going to support these individuals? Where are they going to live? And how is their, what went wrong with their brain? And I personally would like to see, us talk about how we collaborate across all mental illnesses to understand the brain and also to support those individuals living in the community.
So I'm going to start with some questions to open up a discussion within the panel, but I'm happy to take questions from the audience. So I only ask when you have a question from the audience if you could state your name and where you're from. In addition to your question, that would be great. And I think there's probably, I don't know, there'll be a microphone that'll come around. So maybe I'll just start with, we see the prevalence is 1 in 68. It keeps increasing. I think that's a 30% increase since 2008 when they did a big study. And there's a lot of questions now kind of focused on this new DSM definition that takes away the subtypes and focuses on the spectrum, autism spectrum disorder. So maybe I'll start with you, Jerry, and you can talk a little bit about the definition of autism spectrum disorder, the spectrum, and maybe give some thoughts, and maybe Rob too, as to the prevalence, why it keeps increasing. We've touched a little bit on this discussion of the genetics and the impact of the genetics and the environment.
Great. So you may know that there was recently a major change in the way we think about diagnosing autism. Previously, we had many different subtypes. So you've heard of Asperger's syndrome, and that was distinct from classical autism. And basically, those distinctions came from people looking at individuals and deciding that these behaviors seem to be different than these behaviors and putting labels on them. And then over the next several years, scientists tried to validate those subtypes, and they looked to see whether they're genetically distinct, or whether they respond differentially to treatment, or whether they have different neuropsychological profiles, or whether they have different prognostic indicators. And in fact, they did not map onto etiology, treatment, or prognosis, and so they didn't really make sense. The other thing that was discovered in a major multi-site trial where different institutions were making these diagnoses is that even expert diagnosticians were not very reliable at making these distinctions because they're based on pretty subtle differences in behavioral profile. And so because of those two things, it was decided to just do away with those subtypes and to recognize that autism is a spectrum disorder and call anyone who has difficulties in social interaction and communication and repetitive behaviors as being on the autism spectrum. However, we do capture that heterogeneity by the diagnostician making what are called specifiers. So every individual then is also characterized in terms of whether they have comorbid intellectual disability, language impairment, if they have a specific genetic etiology, did they have a pattern of regression, do they have medical comorbidities. And so that's a much more sensible way than of capturing all the heterogeneity that we see in autism. So that was a major shift. We don't actually know when the CDC comes out with its prevalence estimates how this will impact our prevalence estimates. When I was at Autism Speaks in Rob's role previously, we funded a study which should be coming out fairly soon for the CDC to actually put these two diagnostic systems head-to-head and see whether it impacts our prevalence estimates. But Rob, I think maybe, do you want to talk about this notion of increased prevalence and why we're seeing it? That's always the question.
Big question.
Absolutely, and I'll add on that last point. The study that Jerry's referencing is a study called Success, and it's a collaboration that we have with the CDC to use one of their ATOM network sites to not just look at the comparison of DSM-4 versus 5 diagnoses, but really to do that in the context of a different approach to prevalence estimation. And rather than using the CDC's approaches, which rely on really case evaluation, not direct contact with individuals, we've attempted to replicate a study that came out in South Korea that we funded a number of years back that went and did a direct survey of the population in South Korea, and it actually showed a higher prevalence than what we've seen in the most recent CDC estimates. That's due to read out later this year, so we'll have not only a new, potentially a new look at prevalence estimates with another method, but we'll have that in the context of the changing diagnostic criteria. I think there's no question about it that the broadening of the diagnostic criteria has likely contributed to increase in prevalence. There's undoubtedly an increase in awareness of autism that has occurred over the last 20 years, and that's certainly impacted surveillance systems and professional societies introducing into standard guidelines, well baby visits at the American Academy of Pediatrics, etc. All of these, I think, have contributed significantly to the increase in prevalence, but still, there are studies here to suggest that it won't account for everything. And I think anyone who tells you they fully understand what's driving the prevalence probably really doesn't know that we have a clear answer, than that we have a lot of reason to believe these are contributing, but we still can't exclude that there are contributions being made in modest forms by the environment with autism.
Yes, Luca.
There's another potential contributing factor which is still maybe more speculative, but it has to do with the fact that as we learn more and more about genetics, it appears that a large proportion of autism, the genetic of autism, depends on de novo mutations. And de novo mutations are those that are now found in parents, they're in the germline, and they lead to profound effects, obviously, because alone they're able to determine the disease, and they're increasing progressively with the age of the father, so the parental age, and all of this has been determined. Now the correlation between that and the increase in epidemiology of autism is a different story, but there has been an hypothesis that also contributes to some extent.
I think this is a great point. We published a study earlier this year in Nature Medicine, one of the first studies to come out of this missing project that I mentioned earlier. It is one of the largest whole genome sequencing studies to date, where we took several hundred families. These are families that have more than one child diagnosed with autism, and we sequenced both the children and the parents. And going into that study, one might be led to assume that in the case of these quad families, we would assume that more often than not, the children with the diagnosis would share more genetic risk factors in common than not. And actually, we saw the opposite. We saw that more often than not, the siblings both with a diagnosis of autism in the same family actually more often than not had different risk factors, what were presumably pathogenic risk factors present. So that just sort of surprised us all, and one could speculate that could argue for environmental contributions, but I would even say, now that we have this lens of whole genome sequencing that technology is delivering to us, and we have the ability to look at the genome at a resolution we've never been looking at it before, I think we're going to be in for some surprises in terms of how it's organized and how it actually operates. Our assumptions about how it works are probably on the table for challenge.
Can I make one more point?
Sure.
So another, I think, interesting kind of theme that is emerging in the field that relates to this is we have actually identified a number of environmental factors that increase risk for autism. So they include, for example, severe maternal infection during pregnancy, exposure to certain toxins during pregnancy, such as pesticides, and traffic pollution. Traffic pollution has been replicated in multiple studies now. Certain medications that are taken in pregnancy, very early birth in terms of low birth weight, and many more children are surviving now who are born prematurely. So there are all these factors that have been associated with increased risk. And one of the common themes across those factors is this concept of maternal immune activation, right? That these are basically causing an immune activation in the mother, and that we've learned that this then affects fetal brain development. And there's some very interesting research, for example, Stacy Bilbo at Duke University, developing animal models where they're looking at things like stress and pollution and how this affects the animal during pregnancy, and then really has profound impacts on brain development in terms of microglial activation. And many of the early now efforts to try to look at biomarkers for autism that are diagnostic or predictive are really now focusing on immune-related biomarkers. And it'll be interesting to see in terms of even therapeutic approaches that start to really focus more on this immune theme that's emerging.
Yes, we have a question.
My question is for Dr. Ring.
Can you state your name?
My name is Aslıhan Dinser. I'm a PhD student in Mount Sinai Medical School, and I'm working on the epigenetic mechanism in autism spectrum disorders. And last year I got the Autism Speaks grant. So I'm really excited for these missing projects, and I'm wondering, because you are collaborating with the Google research, and how many whole genome sequencing data right now accessible for the public? How can we use it or reach it? So this is one of the first questions.
Congratulations, by the way. I've not had the opportunity to do that in person yet. It's a good question. So we have completed just over 5,000 whole genomes, which is already a massive data set. This is why we're working with Google. The compute power needed to bring onto that is also very significant. I think roughly, last time I looked at it, roughly 2,300 of those genomes have been cleared or are in the cloud and available for access. So you only need to go to www.mss.ng to apply for access, and it's a fairly straightforward process. Again, we expect to complete the sequencing of our target of 10,000 by the end of first quarter next year. It will be a slight delay to get those annotated and fully cleared for use in the cloud. But it's an exciting time. We announced the launch of our portal, which provides researchers that not just that access to the data and in turnkey solutions to analyze the data, but it really brings it down to a command line for the most sophisticated of genetic researchers to draw on all the compute horsepower of Google to go down to the baseboard and do whatever they want with the whole genome files. So.
Thank you. You know, one thing that was really kind of amazing to me is when I was looking at the landscape and looking at, you know, the different types of treatments, behavioral and drug development, and companies that are associated with diagnostic tests, the one thing that was really interesting was this intersection of technology and medicine. And, you mentioned this, Laura, with the interaction you're having with Google and the Smart House, and I know that we've talked a lot about this as well, Rob, and I think maybe if you can just talk a little bit about some of the things that are becoming available and technology, apps that are being developed, and even being developed in a way that they can go through the FDA to get approval. So if you could just, start that conversation.
I would argue this is one of the more exciting areas of the autism space in general. We launched a venture arm of the foundation several years ago called Delcia, and over that transom we've seen more targets emerge in the area of technology than any other segment, if you will, of the emerging marketplace, future marketplace of autism. And a couple of real themes have emerged. Much of that interest we see, the entrepreneurial efforts out there, are leveraging everything from the emerging genetics to some of the fundamental work that's already been done by Jerry and others with EEG and there's eye tracking. The real focus of this is to develop either bona fide diagnostic products that travel down traditional regulatory paths to what one might argue as risk assessment technologies that can be applied in a clinical setting to help triage wait lists, which are a common issue in the autism space. But I would argue one of the more interesting areas that I've seen lately, and maybe it's just a personal interest, but Delcia did invest in a company working on this problem in the last year, is this intersection between gaming, the world of computer games, and the world of cognitive neuroscience, if you will. There's an entire conference called SCONS that focuses on this intersection, but it's a really interesting proposition, the idea of taking tasks that have been used in other settings and put them to work to isolate circuits of the brain like muscle groups in a body to help change performance. There's a company called Achilles Labs that we invested in that is developing a therapeutic game for autism. We help support their proof of concept clinical trial. I know they're working in the area of mood disorders. They're working with Pfizer and with the NIH and schizophrenia. It's a really interesting area, the idea that you could think of an iPad or an iPhone or a computer as a vehicle to deliver efficacy. And Achilles, I think, stands out. Particularly their initial interest was in ADHD, and they had committed to take their development project down the same path as a stimulant might travel with FDA. But I think behind this technology is also an interesting proposition that in these same tasks that that children and adolescents on the spectrum spend a lot of time doing is the ability to collect data about performance. And it may not be too far off that games being run on iPhones and tablets may be the future of measuring the outcome of treatment in the future, or measuring how performance changes over time. So as a biomarker, performance on some of these games is an interesting area. It's certainly something we've been highly interested in investing in as a foundation.
And I think one other question, just to kind of come back to, you mentioned this, Laura, and all of you mentioned this, is the BRAIN initiative. That's a federally funded program. I believe it was 46 to 48 million over the course of, I think it was 5 years. I would just be interested personally to know, scientifically where they're at today. I know that there's been some grants that have been awarded and a committee to define the strategy, but if you could just speak to that, would be good.
I'll make a couple of comments, and I actually wanted to follow up on Rob's and your comment about technology. So Duke is involved in the Achilles trial, so we're excited about that. It's a very interesting concept, and people are even working on similar concepts with infants, if you can imagine that. But to give you another example of technology, we launched a couple of weeks ago at Duke a research kit app with Apple, And so this particular product, it involves a questionnaire that parents fill out, so it's direct to consumer, that is a pretty well validated screener for autism. After they fill it out, the parent also then allows the toddler to watch some videos that we programmed onto the smartphone. And these videos are designed to elicit certain reactions, certain patterns of gaze, but also certain facial expressions. And then our engineering team at Duke has programmed this smartphone so that it can code the facial expressions of the toddler that's watching the video, with the idea that these movements will then help us to increase the accuracy of detection of autism. So really some amazing sort of new ways of thinking about using technology. But just I think the BRAIN initiative is critical. I think, Laura, you really said it beautifully, that across a wide range of conditions, you know, whether it's Alzheimer's or Parkinson's or a wide range of mental health conditions, it is the brain. That is the target organ, and yet we still know so little about it. And the BRAIN initiative is not focused on any particular condition, but it really is development of new technologies that will allow us to probe the brain and understand the brain in more detail. And that's just going to raise the quality of what we can do in all of these fields tremendously. So I'm very excited about it, and you know, some people were upset that they didn't, you know, get funding for their, you know, specific condition that they may have interest in, but I actually think it was a good decision to say we need to invest in the technology that will help, you know, all people that are studying CNS-related conditions to do it in a better way.
And I just want to add that whole concept is really, really disruptive, because so much time and effort has been spent building organizations like Autism Speaks, like NARSED, which has a different name today, for depression and schizophrenia. But the concept of breaking down those walls and coming together to really understand from every perspective, whether it be psychopharmacology, whether it's building beds in the community, whether it's, you know, whatever, to join hands and go backwards and start rethinking, because at the end of the day, just from my layman's perspective, you know, we've been doing a lot of research, but you know, we're not making the kind of progress we need to make, so we need to rethink how we approach this. And I think that's kind of exciting, and I think it's really important to stop and say, hey, should we be looking at this differently and working together instead of in silos?
Yeah, I agree.
Yeah, so important. And I think it goes back, you know, to a point that you always make, Dr. Vagelos, is, you know, the necessity of funding basic research and funding the NIH and these federal funded programs, how important that is. All right. Do we have any other questions? Okay. Oh, we do.
You told us the incidence of autism in the U.S. Can you give us the statistics for Europe and the Far East? Are they similar?
They're similar.
Mm-hmm.
They're similar, and in fact, I think, you know, I've already mentioned a study that we funded in South Korea that would indicate that maybe using even different methodologies, the prevalence may be higher than what we've estimated here. And I think until we've read out and published the study I mentioned, which is being done in South Carolina later this year, we'll have, I think, a real good connection between a global prevalence rate and something done here in the United States.
And I would follow up by saying that we have really fairly good estimates of prevalence. Well, we have good estimates of prevalence based on caseness in the U.S., not as good based on screening a whole population. That's the approach that Rob mentioned that CDC is taking now. We also have good prevalence estimates in Europe and Japan and to some extent maybe Australia, but we actually do not have good estimates, for example, in India. And China
China.
is just working. Right now, the government has funded a very large prevalence study, and that's beginning to come out in terms of publications. But there really are large parts of the world that we still don't have good prevalence estimates for.
Right. I would add, an interesting angle to something I mentioned earlier, but has been, I think, a theme that has prevailed throughout the conversation, is we've never really had a good estimate of prevalence among adults done. And this may itself seem, why would we do that? Well, in addition to collecting some other vital information that would come through a study like that is a black hole for us, it may help us get at that question of whether or not prevalence has truly been changing over time, if we could actually understand what that prevalence is in the different age groups. And I know that this has been talked about. It's certainly an area we're quite interested in funding a study in, and we may have some traction on that in the near future. But that is, I think, a gap for us in the field right now.
Okay, well, as we come up on the close of the hour, one more question? Okay, and please state your name and where you're from.
Sure. My name is Vered Biskerleib, and I work for Sidon Development, which is an orphan drug accelerator. And my questions are to Dr. Santarelli and Dr. Dawson. You mentioned the studies with abaclofen from Seaside and the mGlu-R5 from Novartis that have been disappointment. And there was a lot of criticism about the studies when they stopped for fertility, and whether the right endpoints were really taken into account. And my question to you is, what do you think should be really the right clinical endpoints for an autism spectrum disorder or Fragile X kind of syndrome study that would be meaningful and clinically meaningful and accepted by the FDA?
So, I'll take that. Okay. So I don't think we know what they should be. Otherwise, everybody would have done it already. We're still the reality, we're still learning. And what I said before we're, and just to quote the former CEO of Seaside, we're kind of building a plane while we're flying it. And so you end up with a molecule that has interesting profile in an animal model, and it all lines up. It looks like it should work in humans. And then you face in the clinical complexity, which even if you're in monogenetic disorders, you have different level of severity, a gender influence, you have an age influence, and you're trying to manage all of that. On top of that, there aren't way of measuring quantitatively improvement that are validated yet. And the only way to validate them is using a drug, because or perhaps behavioral treatment. And so you really, is it your typical catch-22 situation where you're really trying to learn iteratively, and at the same time you're working against a patent lifetime for a particular agent. So all of that is the challenge. So we don't have an answer to that, but the way we are approaching it is trying to reduce as much as we can heterogeneity, and trying in all those ways that I've told you. And one way is to choose a targeted genetic condition, so at least we know there's one cause only, or a predominant cause, and then trying to stratify as much as we can to reduce the noise in the clinical trials. But until you do that well, it's impossible to discriminate whether it's the target the problem or is actually the design of the clinical trial.
Just briefly, so the arbaclofen trial with Seaside, I actually went to the FDA with them to try to argue that to try to salvage that trial, which we were not successful in doing, because there was a subgroup of individuals that appeared to have a true signal, but of course it was post hoc, and that's not acceptable. But it does point out this idea of how do we identify the right population. So I think this investment by the NIH, $34 million, so it will be a cohort of 250 4 to 11-year-old children that are evaluated at baseline, 6 weeks later, and 6 months later, using a variety of parent report forms, clinician-rated forms. We're checking 7 EEG paradigms, possible biomarkers. 10 different eye tracking paradigms will be examined. And we're looking at things like test reliability and how these biomarkers correlate with parent report measures of social communication. And the entire thing is to try to come away with a set of measures, EEG, eye tracking, parent report, and clinician, that converge and are highly reliable and sensitive to changes over time. So it's a 4-year project and 5 universities, and we hope that at the end of that we can come away with some answers to the field. And I will just say that our baclofen trial took a questionnaire that was developed for individuals with intellectual disability, not autism, that was developed many, many years ago, and took out a subscale of it that was on social behavior, and that was the outcome measure. So, that is just such a crude tool to try to measure changes, and it just doesn't have assay sensitivity. So we're very excited that the NIH project may move the field forward and allow pharma to be able to conduct more successful clinical trials.
Okay, I'm going to take, I guess, 2 last questions.
Sarkis Mazmanian from Caltech. So we've spoken a lot about the behavioral abnormalities in autism, which are the core defining features, but there are also certainly comorbidities and a spectrum of comorbidities with autism that range from seizures to sleep disorders, metabolic disorders, gastrointestinal complications. Are there therapies, or at least a pipeline or thinking about addressing these complications, these comorbidities, because they could provide improvements in the day-to-day lives of these children with autism?
I'm happy to start with that. This is one of the myths that I think has to get broken through by many who look at autism for the first time, that, yes, there are these core features, these cardinal features that most associate with autism, but the clinical reality is much more complicated than that. There's an entire, spectrum of associated comorbid conditions that you've already described, and each one of these might be considered a target for therapeutic development. In fact, one might argue, at least we did well at Pfizer, that these are leveraged areas that have already been prosecuted: anxiety, sleep, seizure, and the challenges there may be found in some of the regulatory paths, the charges of pseudo-specificity that one might encounter along that way. But I think in reality, these are very tractable areas. In fact, the 2 approvals that we have in the space to date are Risperdal and Abilify, which target a cluster of associated symptoms called irritability, which include self-injury and tantrums and aggression. So there's already a precedent set by that. I think the reality, for those of you new to the space, this reality of autism as a unitary concept is already failing beneath the weight of what we already know. And in reality, autism will likely play out like cancer and be described by a spectrum of hundreds, maybe, of different autisms. And it's going to be through the lens of efforts like genetics that help us create that ontology of what autism of the future will be defined. And I think before even those pay out, we already have this clinical reality of the associated symptoms to help us create that ontology. And I think that's a tractable area for medicines developers that may not have been fully explored.
So thank you so much. I think we have to, I'm being told we have to close. We have to leave the stage. But thank you for your attention. Thank you, the Galien Forum. And I'm sure the panelists are available to speak with you further. Thank you so much.